Hello everyone, welcome to Gebze Technical University Department of Bioengineering webinar series. Our guest for today's webinar is Dr. Gülsen Betül Aktaş from Bilgi University. Dr. Gülsen Betül Aktaş was completed her bachelor degree at Ege University Department of Chemical Engineering, and she completed her master in Department of Nanoscience and Nanotechnology at University of Rovira Virgili. During her doctorate and postdoctoral studies, she worked at same university, Department of Nanoscience, Material and Chemical Engineering. Currently, Dr. Aktaş has been working as academician at Bilgi University. Dear doctor, stage is yours.
Thank you so much. Yes, I hear back my voice. Is it clear, my voice, by the way?
Yes.
Perfect. By the way, do we continue in Turkish or English?
You can prefer.
Okay, I will continue in English then. So today I will give a presentation. Let me share the presentation.
Okay, we see.
And I need to see in a couple of seconds. Perfect. Okay, perfect. Is it clear?
Yes.
Perfect. So today I will give a brief presentation what I have done in my PhD basically, and afterwards the work that we have done. And the title of the presentation that I will be giving today is the application of DNA binding protein tags as means for defined co-immobilization in biosensor. So for this purpose, I will give a brief introduction, then result and discussion of 4 different parts of the project and the conclusions that we have obtained from these projects. So as many of you know that biosensors basically are analytical devices that converting a biological response into a signal, and we are using widely in different applications such as medical diagnostics, food safety, environmental monitoring, biological warfare agents detection, etc. So a biosensor basically is consists of a surface that you're attaching, that your bioreceptors, and then the analytes that you need to capture with the bioreceptors, and then from this capturing or afterwards a signal that you are providing, that you are reading out if you have the disease, if you have the diagnostics, etc. So basically, mainly we are generating the signal with 2 different methods, label-free and label-based in biosensors. Label-free has the advantage of not using any label, so you don't need to do any labeling process for your biosensor. So you can directly detect your target. And some of the applications of the label-free is basically electrical techniques. You can use the surface plasmons, resonance, mass spectrometry, etc. They have the advantage of not using the labels, but also they have the disadvantage of using expensive instruments, and as well as you need trained personnel. And on the other hand, you can use label for your biosensors, and these ones that the labels that you can use are fluorescence, nanoparticles, enzymes, and they provide you low-cost instruments because you do not need a huge instrument to detect your target, and you can basically detect with naked eye. So you don't need to do you don't need to have any even additional instrument to get your signal, and these make your biosensor point of care. Okay, so some of the examples of the labels basically fluorescence, and how the fluorescence is working with the light that it gets excited, and from this excitation you get your emission, and you know with this light if you have the targets on your biosensor or not. in this case, if you use the fluorescence, you do not need any substrate, additional substrate, and it provides you sensitive detection with this emission and excitation of the light, and only light source is enough to read out of your signal. And in the case of the enzymes, they are very, very good to generate the signal and amplify the signal. Basically, the enzymes is catalyzer of your system. And if you have the enzyme, when you provide the substrate, it's gonna go catalyzing, so you will amplify your signal naturally. And they are specific because they work with the key and log system. They are specific to your substrate. And in this case, they are providing a specific and sensitive detection. Another labeling method for the signal generation is basically using the nanoparticles. Why we like the nanoparticles? Because they are very easy to prepare nowadays. So you can prepare your gold nanoparticles, carbon nanoparticles, magnetic nanoparticles, different types of the nanoparticles easily in laboratory. And they are stable, so they are not like enzymes that they will have an expire date. Also, they provide you a large immobilization surface that you can attach different biological molecules. With this, you can increase the signal, and you can have a strong signal to read out in the end of the biosensor design. So what we know, for all this approach to assemble a biosensor, you need to immobilize protein. Either you need to immobilize protein on the biosensor surface, like you can immobilize antibodies, enzymes, streptavidin, or you need to immobilize for detection. You can immobilize your proteins on the surface of the nanoparticles, or you can immobilize directly for the detection probe. And what are the methods, common methods for the protein immobilization? Is the physical absorption, the covalent binding, or bioaffinity interaction. So for example, the protein immobilization is using the different type of the intermolecular forces like hydrogen bonding, van der Waals forces, electrostatic forces, hydrophobic interactions. They have the advantage of being very simple. So you just throw your proteins, you will have immobilization of the proteins, and it provides you a low cost because it doesn't require any additional step. But what you have here, a random orientation, and you cannot control how they are going to be binding on the surface. And also by time, you will have the protein desorption because it's not covalently linked. It's just basic interactions. And by time, this interaction will disappear, right? And in the case of the covalent linking, you can use the side groups of the proteins like lysine, cysteine, aspartic acid, glutamic acid. And here, the advantage of the covalent linking, you can control the immobilization because you know the site groups, right? And usually is irreversible because it is the covalent linking. Even you want to break down, you need to use another chemistry. But still, because your protein or any protein will have can have multiple sites on its surface, you can end up on the random orientation. And this can result in the protein inactivation. Because here, for example, you have the active site, and when you have the site groups here, it can be immobilized close by, and this will decrease the protein this will decrease the inactivation of the protein, and this then will result on the decreasing the signal on the biosensor. Another approach for the protein immobilization is using bioaffinity introduction. One of the very well known is using the streptavidin biotin because it has very, very strong binding. And the rest that we can use is the antibody antigen. We can use the protein A and the FC region of the immunoglobulins, and we can use the polyhistidine tag and the metal ion. What is the advantage of that? It provides us the strong interactions and it's specific. So it will be only bind when it has the coupling of this interaction, the bioaffinity. but still you can have the random orientation because you don't have always like 1-to-1 ratio for the interactions. In the case like streptavidin, biotin, streptavidin has 4 binding sites for the biotin, and you cannot control where it's gonna binding. And in the end, you will have a heterogeneous surface coating. So our approach for the protein immobilization as an alternative bioinfinity interaction is using DNA binding proteins and double-stranded DNA. So what are the DNA binding proteins? DNA binding proteins are the proteins that have various in vivo functions related to DNA. They are in the function of the transcriptional regulation, DNA processing, DNA packaging, etc. And DNA binding proteins can bind single-stranded DNA or double-stranded DNA specifically or non-specifically. So our main objective of this work was to develop a new immobilization system based on the interaction of DNA binding proteins with double-stranded DNA and apply them on the biosensors. So the first part was basically immobilizing the DNA on the biosensor surfaces using DNA binding protein approach. So mainly what we use nowadays is using the streptavidin biotin interaction for the DNA immobilization on the biosensor surfaces. So what you do is basically first you immobilize your streptavidin, and then you introduce your biotinylated DNA, and because it has very high affinity, it's gonna bind on the streptavidin coated surface. You will have your DNA immobilized on your biosensor surface. As an alternative to this, approach, what we can do, we can use the DNA binding protein and double-stranded DNA sequence. So what we can use a specifically binding DNA binding protein. One of them is the single-chain version of the bacteriophage chlororepressor protein. What is this protein? It is a monomeric protein. It is very small, 17 kilodalton, and it is very positively charged, which is logical because DNA binding protein is positively charged, DNA is negatively charged, and you will have this interaction. But it binds very specifically to its sequence that has this sequence. It only recognizes this sequence and is going to bind to this DNA. So what we thought, we said, if we have these DNA binding protein sites for the single chain CRO, we can use it to immobilize the DNA. So what we can do, this is the DNA part, single-stranded DNA parts to immobilize, the part that we want to immobilize. And this is the part, this double-stranded DNA that has single-chain CRO binding site. So if we immobilize single-chain CRO on the biosensor surface, and after that we introduce our designed probe that has the binding site plus the part that we want to immobilize, we will have in the surface the DNA that we want to immobilize. And we can compare this immobilization with the streptavidin biotin approach. What we have done for this, basically, we designed our single-chain CRO DNA binding protein with addition of the N-terminal cysteine and C-terminal histec. And why we did that? The cysteine to be reactive, and the C-terminal histec to purify it, easily purify it. After that, we express it and we purify it, and we obtain total yield of 2 milligrams per liter of the culture. And this is the purification level, more than 90%. And what we do in the biosensors, one of the approach is the protein immobilization using the absorption technique. So if you have a strong surface and protein interaction, even you wash it after time, it will remain on the biosensor surface. But if you have a weak surface protein interaction, what will happen after the washing and the time steps, you will lose some proteins on your biosensor surface, and this will lead you a non-sensitive target detection. Why? If you have more immobilized protein remains on the surface with the stronger surface interaction, you will have more protein, and then you will have more sensitive detection. So what we did in the beginning, we basically poured the streptavidin and single-chain CRO on the biosensor surface, one of the biosensor surface, which is nitrocellulose. And after that, we wash them either with water or PBS twin, and then we paint them with the, basically we stain them with the comestible. And what we have observed when we use equal mass of the streptavidin and single-chain CRO, or equal molarity of the streptavidin and single-chain CRO, when you wash with the PBS twin, streptavidin vanish away. I mean, they go away from the biosensor surface more than the single-chain CRO goes away. After we have seen that, we said, okay, let's check in the full configuration of the biosensor. Let's use the ELONA, which is enzyme linked oligonucleotide assay. And here, for this approach, either we immobilize with streptavidin or single-chain CRO on the biosensor surface, or either with the biotinylated single-stranded DNA capture probe, basically, or our design single-stranded DNA, double-stranded DNA. And then we have our target, which is high-risk human papillomavirus, HPV16, single-stranded DNA, and then the detection probe that has HRP. And we use these two configurations to compare them. And what we have obtained as the signal, this is the target concentration, increasing the target concentration. When we use the single-chain CRO, the same amount of the single-chain CRO or the streptavidin, we obtain 5 times more sensitive results when we use the DNA binding protein. And what we have seen, we can decrease the limit of detection of the biosensor if we use the single-chain CRO DNA binding protein interaction. After that, we have seen, okay, this is one technique, enzyme-linked oligonucleotide assay. Let's move on to lateral flow assay. And let's check on the lateral flow assay if they provide you a sensitive signal. We herein we check the same molarity or the same mass in the milligram, and we have seen the single-chain CRO provides a better signal than the streptavidin, even in the same molarity. And why it could be? Why we said, okay, the single-chain CRO provides us a better signal, but why? Most probably is because it's super positively charged. The pI of the protein is around 9.6, and the streptavidin, on the other hand, is kind of acidic, 5 to 6, the pI. And the nitrocellulose and the polystyrene microplates that you are using on the biosensors, they are treated, they are negatively charged. And because of this positively negatively charged interaction, single-chain CRO remains on the biosensor surface more stronger, and we improve the absorption basically using this high positive charge of the protein, thanks to this high positive charge of the protein. And hereby we increase the signal of the biosensor, and also we get rid of using the biotinylation, which makes the capture probe much cheaper. So what we get in the end of this first part, we developed single-chain CRO, a new protein for the immobilization of the DNA, single-stranded DNA. And basically this system based on the interaction of double-stranded DNA and specific DNA binding protein. And here we do not need any chemical modification, like biotinylation is basically oligonucleotide, which makes it much cheaper. And we improve the signal in both single-chain CRO compared to streptavidin in nitrocellulose and polystyrene microplate, which are two commonly biosensor surfaces. And we can explain this improvement by the positive charge, high positive charge of the single chain CRO. And this work has been published on analytical and bioanalytical chemistry. And in the second part, we get our DNA binding protein and we get the single-stranded DNA detection probe, and we wanted to generate the signal generation by using one DNA binding protein. So we have seen that we are using biosensors a lot, and they are very important for the diagnostics, and we have the genosensors in the world of the biosensors. What are the genosensors? Genosensors are basically the biosensor detecting the nucleic acids. They are widely used in medical diagnostics, gene expression analysis, detection of the infectious disease, biological warfare agents. And the classical configuration of the genosensors is basically you are capturing your target DNA with the capture probe, and then you are detecting with a detection probe that has the label. And some of the examples for the signal generation in genosensor, either you use functionalized nanoparticles, and you can use the reporter enzyme level like HRP. But we said the DNA, we can immobilize the DNA on the nanoparticles, and you can immobilize many of them. But they have the size and distribution issues, and you need to modify all batch again and again on the nanoparticles when you change the target. So if you change the target, you need to immobilize different single-stranded DNA to capture it. And in the case of the enzyme, again, you have the same problem that you need to design again your HRP with different single-stranded DNA per target for each target, basically. And here, again, you have a limited signal. Why? One target, basically, you will have one HRP. So one target when you will get one signal. So what you need, basically, you need a higher signal amplification than single-stranded DNA HRP. So you need to increase your HRP per target molecule, but with using a better reproducibility than functionalized nanoparticles. How you can do that? You can use DNA binding proteins by attaching more reporter enzymes per target molecule. How you are going to attach more reporter enzymes per target molecule? Basically, you can use the DNA binding proteins and their sites. You know where they are going to bind, and you know how they are going to bind. And by knowing all this, you can design your detection probe that has more reporter enzymes. How you are gonna do that? You will get the single-stranded, single-chain CRO DNA binding protein, and you can conjugate it with the HRP. In the end, you will have your complex DNA binding protein and HRP, and you will design your detection probe that has binding sites of this DNA binding protein. And you can design them to have face-to-face, not to have any steric hindrance effect. So they will be freely binding on your detection probe. And when you introduce your the complex DNA binding protein HRP complex, when they see there is this sequence over there, they are gonna bind, and you will get your signal. How you are going to design that? You will have a hybrid single-stranded DNA, double-stranded DNA detection. Single-stranded DNA part is basically to detect the target, is going to hybridize on the target. And double-stranded DNA that will have multiple DNA binding sites with specific sequences. So what we did as an approach, we get the classical configuration which has the detection probe that has one HRP, and our detection probe that has single-stranded DNA that gets detecting the target, and the double-stranded DNA that has multiple binding sites. And to get the conjugate of HRP single-chain CRO, we reacted them with this, because it has the N on N-terminal on cysteine, and the HRP has the maleamide, they got reacted. And we checked the reaction after the reaction with the STS page. We have seen that this is the size of the single chain CRO. This is the HRP. After that, we have purified with the IMAC and using the concanavalin A, and we get our HRP single chain CRO. And then we wanted to see how many HRP molecule we have per single chain CRO, because we know single chain CRO has one cysteine, but we do not know one HRP molecule averagely how many maleamide it has. After that, we checked with the multitoph, and we have seen that we have the conjugate containing 1:1 ratio of single chain CRO and HRP. Also, we get to our conjugate, but we wanted to see, is it still binding to DNA as expected? And we introduced our DNA that has one DNA binding site, and then we introduced our detection probe with 3 DNA binding sites. This is the free DNA, and this is when we add the HRP. HRP doesn't bind to DNA as expected. And when we introduce the single chain CRO, there is a shift due to the binding. And when we introduce our HRP single chain CRO conjugate, there is again a shift. And here you see the shift 3 times because it has 3 DNA binding sites. Well, some of them binds 1, some of them binds 3, some of them binds 2. And we confirm here the binding of the single chain CRO and the HRP single chain CRO specifically comes from the single chain CRO. Then we move on the detection of single-stranded DNA using the ELONA. Basically, the classical configuration, our detection probe that has 1 binding site, and then our detection probe that has 3 binding sites. When we have worked on these 3 different configurations, what we obtain the similar result, basically the same results. We have the 1 binding site and the classical configuration that has the detection probe HRP. And when we introduce 3 DNA binding sites on the detection probe, we increase the signal 3 times. The slope, basically sensitivity, increased 3 times. Then limit of detection decreased 3 times. So what we have seen here, by using the DNA binding protein, we can control how many HRP molecules we can immobilize on the detection probe, and we can amplify the signal. So per 1 target, you will have more than 2 times, like basically up to 3 times of the signal. So you will have early diagnostics for your target detection. Then we check the specificity of the Biosensor. We introduce different targets, basically CDH1 or HLA, and our Biosensor was specific to our target. So what we have seen here, we can make a novel HRP DNA binding protein conjugate, and this conjugate only binds to double-stranded DNA with the specific binding site. And with this, we successfully detected our target using hybrid single-stranded DNA, double-stranded DNA detection probe that contains specific binding site. And we increase the sensitivity compared to the classical configuration of HRP conjugate with the DNA, single-stranded DNA. And this is universal conjugate, because when you change only the single-stranded DNA parts, double-stranded DNA will remain the same. You can use for any target, any DNA target. And this also decreases the cost of the assay because it's universal. There is no any chemical processing like biotinylation, like HRP molecule addition on the single-stranded DNA. You just need to basically increase your detection probe with the oligonucleotide synthesize, and you can use it for any type of the target of the DNA. And this work has been published on biosensors and bioelectronics. And in the second part, what we did, we generate the signal using 2 DNA binding proteins instead of the 1. So what we know, the HRP enzyme is typically signal generation element in Biosensors and is using hydrogen peroxide as a substrate. But hydrogen peroxide is not stable. So you cannot use for point of care assays. What you can do instead, you can add a glucose oxidase enzyme nearby. You can generate your hydrogen peroxide by only adding the glucose, which is a stable molecule. And glucose oxidase enzyme has a rapid signal generation for glucose. It has a very good enzyme stability. Also very high turnover numbers, which means very active enzyme. And you can have an enzymatic cascade with HRP. So if you introduce your glucose oxidase, you can basically oxidize the glucose in situ. You are going to generate the hydrogen peroxide. What you can do with this cascade, enzymatic cascade, a point of care biosensor. How we can do that? We can use a secondary DNA binding protein to conjugate with glucose oxidase and use its different specific double stranded sequence. And hereby you will have your enzymatic cascade, single chain HRP, and secondary DNA binding protein that has conjugated with glucose. When you introduce your glucose oxidase, when you introduce your glucose, you will produce first hydrogen peroxide, and this hydrogen peroxide will be catalyzed by HRP. For the conjugation of glucose oxidase, here we choose a headpiece domain of Lakai repressor protein from E. coli. Again, it has high affinity and specificity to double-stranded DNA, and is a single chain dimer that has we introduced the N-terminal cysteine. Again, is small in size. Again, is binding to a specific, different than single chain CRO, different sequence, binding to double-stranded DNA. So we again prepare the glucose oxidase headpiece conjugate overnight reacting with the cysteine and maleimide glucose oxidase, and we purify it. And we checked again in Maltitov, and we have the glucose oxidase headpiece conjugate 1-to-1 ratio. And we designed our single-stranded DNA, double-stranded DNA conjugate. This basically the detection probe, having different ratios of DNA binding proteins, either 1-to-1 single-chain CRO headpiece, or 2-to-1 single-chain CRO headpiece, or 1-to-2 single-chain CRO headpiece. Why we did that? We wanted to see which one providing us the best signal. So this most sensitive signal for our biosensor. And we checked the binding of headpiece and CRO simultaneously, and we have seen the shift, the bound DNA. And this showed us the simultaneous binding of 2 DNA binding protein conjugates obtained on the detection probe. We optimized our ELONA, we optimized the capture probe concentration, the detection probe concentration, enzyme conjugate addition order. If we add the glucose oxidase first, or if we add the HRP first, their conjugates, or simultaneously. And from these results, we get the optimized condition, and we move on the detection of single-stranded DNA targets. And here we obtain the best result when we have 1-to-2 glucose oxidase HRP molecules. So this explains that if you have excess of glucose oxidase, you are gonna increase the assay sensitivity. And here again, we could control where to bind, how many molecules, how many enzyme molecules that you want to bind. And here the target was HPF16. We checked again the specificity of the Biosensors with different targets, and we obtained our results with our targets only. And we conclude here we can have a second enzyme DNA binding protein conjugate, which is glucose oxidase head piece. And this can be used with HRP as an enzymatic cascade to generate hydrogen peroxide in situ. So you can have a point of care biosensor. And here we can play with the ratio easily by playing with the specific binding sites on the detection probe. We can increase the sensitivity and improve the limit of detection using the glucose oxidase in excess. Again, they are universal conjugates, and this time you have a suitable for point of care testing on the biosensor. And this work has been published on bioanalytical chemistry. On the third part, we have directly detected the PCR product using DNA binding proteins. So we have the lateral flow assays, and this became super popular. Even nowadays, you know, the fast test for the coronavirus, after the coronavirus, and commercially is already available with the pregnancy test. And why they are so popular and also reachable, because they are very simple and they are cheap, because you are basically using a membrane that has paper, and they are user friendly. You don't need any equipment to detect your target, and they provide you sensitive signal. And mainly, how is the configuration behind it? You can immobilize the antibody on the nitrocellulose, and then you have, in the case of the nucleic acid, you will have the double-stranded DNA that has here the antigen, and again here the biotin, for example. And when you introduce your nanoparticle that has the streptavidin on top of it, you will have immobilization. And for this approach, what you do, you are introducing on the PCR your labels to get these labels on the beginning and the end of your DNA. Basically, you are putting your primer that has label, and your primer in the end of the primer that has second label. And then in the end of the double-stranded DNA, you will have that has labels. Our approach, instead of using the label, introduce the DNA binding sites on the primer. So after you run the PCR, you will have your target, double-stranded target, that has DNA binding site. And here, why it's important? Because again, the primers that has this specific here, they are increasing the cost. If you eliminate, for example, Alexaflor, or if you eliminate any other type of the antigen, you will decrease the assay cost. And we used as a target the Shiga toxin producing E. coli. And what we did, we said, okay, we can use our DNA binding protein on the immobilization carbon nanoparticles. We compare them with the HRP single chain CRO conjugate, which one will provide us the better signal, and HRP single chain CRO, both the carbon nanoparticles, even to enhance even more the signal, enhance the signal more. So before we are move on to lateral flow assay, we checked the compatibility of our PCR product. We run on our PCR, we introduce our primer, so we get our PCR product with the target sequence and then the single chain CRO binding sites, either one binding site or either two binding sites, and we checked on ELISA. And we have seen in ELISA that it perfectly worked. We even increased the signal, detection signal, when we use the two binding site of the CRO. And what we did, we immobilized, after we have seen that it's working, we immobilized our DNA binding protein, the single chain CRO on carbon nanoparticle, or our DNA binding protein HRP conjugate on carbon nanoparticle. How we did that? We used this physical absorption basically, and we washed after this physical absorption to get rid of any unbound protein. And we checked on lateral flow assay using different DNA concentrations, increasing the concentrations. Here you can see very clearly the two, when we use the two binding sites, you have a better signal, visualized signal, and also very clear signal when you increase the target concentration. And when we have used, instead of the carbon nanoparticle single chain CRO conjugate, when we use CRO HRP conjugate, we even get a better signal. Why? Because it's enzymatic reaction, and it provides already is generating the signal and increasing the signal instead of the carbon nanoparticles cannot amplify the signal, but enzymatic reaction can even increase the signal. And when we used the HRP single chain CRO with the carbon nanoparticle, both of them, we even enhance the signal more and more, and we got 2x when we have the two binding sites, a better result as well. And we said, if you want, for example, a sensitive signal, you can use this approach. But if you want to make it simple and no enzymatic reaction, you can use direct the carbon nanoparticle with the DNA binding protein. Or if you want directly the conjugate, you don't want to immobilize on the carbon nanoparticle, you can use directly the enzymatic reaction by carbon nanoparticle, without carbon nanoparticle, HRP and single chain CRO. So here we described the lateral flow assay, with the use of the single chain CRO and HRP single chain CRO, and single chain CRO binding site we added by using the PCR, basically with the primer in the amplification step, which was very, very easy. And we demonstrated 3 different strategies. If you want to use, if you want to obtain very sensitive, you can use the HRP single chain CRO conjugate with the carbon nanoparticle. If you want the simple, as I explained, you can use the only carbon nanoparticle. And you can use, depending on your approach. And also we decreased the cost of the assay because we eliminated one of the label on the PCR step. And this worked. The result has been published in Microchemical Acta in 2019. Afterwards, we move on. Okay, we said we are detecting all the time the DNA. We are using the DNA because we know the single chain CRO or headpiece is going to bind on the DNA. But can we detect, instead of the DNA, can we detect protein using DNA binding proteins and aptamers? So, what are the aptamers? Basically, aptamers, biomolecular recognition elements. They are like antibodies, but in a cheap way, and they do not have any batch-to-batch variation like antibodies. They are very easy to modify because they are basically oligonucleotides, and they are flexible. You can play with them. They are not gonna be denatured irreversibly like antibodies, and you can denature them reversibly basically. And we already developing so many aptamers because they have that much of the the advantage. So even we have some aptamers that have dual aptamers, we call them that, like for example, one of the example is thrombin. Thrombin has 2 aptamers that binding to different sides of the protein. So what we said, we can detect the protein by using the aptamers and our DNA binding protein conjugate. We can use dual modified aptamers for sandwich detection, and we have seen the HRP glucose oxidase cascade system is working for signal generation. So let's move on using the thrombin as a model target protein. So thrombin is going to bind to its aptamer. Then we are going to design an aptamer that has binding site, different binding sites, either with the single chain CRO or headpiece. And let's see if we can detect protein this time. And how we designed our aptamers for this approach. Basically, we have our thrombin binding aptamer. This is universal. This is already well known. And then we introduced a spacer, not to have any steric hindrance effect. And then we designed a single-stranded DNA, but it's complementary to itself. So you will have hairpin in the end. And this hairpin is going to be a double-stranded DNA. And this double-stranded DNA sequence will lead you to binding to your DNA binding proteins. So what we did, basically after this designing, we checked their binding, because we don't know, okay, the thrombin is binding to its site, but what happens if we design our aptamers and thrombin binding is affecting to our binding to DNA binding protein, or DNA binding protein is affecting to our thrombin binding site. So we check them simultaneously. We introduce them to their, this aptamer, thrombin, and DNA binding proteins simultaneously. And we also introduced our conjugates simultaneously, and we again have seen the shift, which we were happy is simultaneously binding, so we can detect. And we optimized our thrombin detection using capture aptamer concentration, different capture aptamer concentration, different detection aptamer concentration, and then enzyme conjugate concentration. And with all these optimized conditions, we move on the detection of the thrombin. And because we knew from beginning that if we add glucose oxidase more, we are obtaining a better signal. And we check that if we have 1 glucose oxidase or 2 glucose oxidase, and when we have added the 2 glucose oxidase, we increase the sensitivity. Very happy, because it's very easy, just addition of the glucose oxidase headpiece binding, basically headpiece binding site, you will have more glucose. And here what we have seen, we can not only detect the DNA, but also detect the proteins using DNA binding protein approach. And we can easily modify the aptamers, and we can have hairpin configuration, and you can use for different aptamer targets configuration. So not only the thrombin, but if you have different target that has the aptamer, you can modify it universally, you can use it. And it's very easy to modify the aptamer, again, because you are just basically increasing the sequence, the oligonucleotide sequence. And again, it has a potential of using the point of care, because you are not using any substrate that is not stable. You have your glucose oxidase over there, you have your HRP over there. So what you will have, directly detection with the glucose. So this work has been published on analytical and bioanalytical chemistry. And what we have seen in generally in all the work, we can develop a co-immobilization system. In this co-immobilization system, we can control how many enzymes, molecules you want to immobilize on your biosensor, and how many of them you want them to be more active on your either detection probe or your capture. This system is very suitable for biosensor applications, especially if you are using for the detection of the DNA, but not only the DNA. As I said, you can detect the proteins by using this approach, and this makes for the systems cheaper, which you will have a lower cost assay universally, and you can also have directly point of care system. And what we can do in the future, we can perform lateral flow assays using this time to both sides, both capture and detection probe without any other labeling system. So we can both eliminate the labels in both capture and the detection, which are even further gonna decrease the cost of the assay. And we can identify even more DNA binding protein candidates to expand the what we have until now. And if we have more DNA binding proteins, we can explore multiplex biosensor applications, and we can do multiplexing different targets at the same time. And also we can check what we can do more with these DNA binding proteins and other potential applications using. And this work has been done with the mercury actions with 70 frame program at Rovira Virgili University in the Interfibio group, and also with the Wageningen University, Netherlands, and with the help of the short-term fellowship with EMBO. Thank you for listening, and if you have any questions regarding the presentation, I am happy to hear that.
Thank you, Doctor. Thank you for your impressive presentation.
Thank you.
If there is any question about the presentation, please ask right now. I suppose that there is no question.
If not, you can email me anytime.
Okay, thank you again.
Thank you too.
Okay, see you later.
See you later. Hope you have a successful time at Gebze University and joyful laboratory times to everyone.
Thank you so much.
Thank you. Have a nice day.